Today we shall revise from our chapter number 5 the following important points, like number 1 cytoplasm, number 2 nucleus, number 3 cell division, cytoplasmic organelles like endoplasmic reticulum, Golgi bodies, lysosome, mitochondria, ribosome, vacuoles, and at the last we will see how the plant cell differ from the animal cell. All these points today we will revise. In the earlier class of revision, we have studied everything which are outside the cell, like the cell membrane, like the cell wall, and some important molecules activity which includes the movement of the molecules like the osmosis, diffusion, etc. And we have also finished in our revision class the different discoveries event in the history of cell. But today we will enter inside the cell. What are the things which are present inside the cell? When we will talk about the animal cell, then after plasma membrane or after the cell membrane, what will be inside the cell? And similarly, when we will talk about the plant cell, then we have also what are the content within this cell and what are the content of within these cells. This is the plasma membrane or the cell membrane, and this is the cell wall or the plasma membrane. Both are present within the plant cell, and only a single surrounding membrane is present in the animal cell. But first we shall talk about the cytoplasm. You have seen that within this membrane bonded structure there are matrix or fluid substances within which there are presence of this nucleus and the cytoplasmic organelles and the other non-living substances. We will discuss all things. Just within this membrane there are presence of matrix or jelly-like or the fluid substances which is the place for the nucleus, which is the place for the other non-living substances, which is the place for the different living cytoplasmic organelles having the different structure and having the different functions. Now first we will look about the cytoplasm. Now look here, what is cytoplasm? Cytoplasm is the fluid portion of the cell present between the plasma membrane and the nuclear envelope. If you have an idea about the structure, then like you may have seen that this type of structure here there are presence of chromatin network, there are nucleolus without any membrane and nucleoplasm. This is the structure of the cell, and this the rest part is besides nucleus. All these portions are the fluid portion of the cell present between the plasma membrane. This is the plasma membrane and this is the nuclear envelope. So between the nuclear envelope, you may look here, the nuclear envelope and between the plasma membrane. These are the plasma membrane. This is the plasma membrane, and this is the nuclear envelope, and may you have see here there are presence of this matrix. So this matrix, which consists of the three important part. Number one, cytoplasmic matrix. What is this cytoplasmic matrix? That is also known as cytosol. Understand? Cytoplasmic matrix or cytosol is a jelly-like semi-fluid within which there are presence of living cell organelle like mitochondria, like ribosomes, Golgi complex, lysosomes, etc., plastid. All these living cell organelle having the different structure and functions are embedded, are present within, are floated within this cytoplasmic matrix or cytosol. And there are also presence of the non-living substances, that is known as cell inclusions within this cytoplasmic. You well know that each and every functions which are related with the life are performed within this cell. That is why the different types of cell organelle and also the different type of substances which are present within the cytoplasm perform and help in the different vital activities of the life. May you see all those functions separately when we will study how the protein synthesis, how the process of biogenesis, that means the for the formation of the cell membrane there are requirement of the lipid, there are requirement of the protein, how that will be fulfilled with the help of the endoplasmic reticulum. And similarly there are the releasing of energy that will performed by the mitochondria. Within the plant cell there are presence of plastid which possess the chlorophyll molecules that will helpful in the process of photosynthesis, and there are a lot of functions, all the functions, and also there are presence of the nucleus within this cell that will perform vital functions. All the activities, metabolic activities will be operated or performed with the help of this nucleus, and also there are presence of the genetic material within this nucleus. That's here the first you have seen here the cytoplasmic matrix, and the second one the cell organelles, and the third one non-living inclusions. This is the structure, the summary of the cytoplasm. Now look here, the different non-living substances which are present within the cytoplasm. The cytoplasm contains various inorganic molecules such as water, sodium, potassium, salts, and various organic materials like carbohydrate, lipids, proteins, nucleic acids, and several enzymes. Whatever may be the requirement for the different activities which are related with the life, or whether it is the requirement for the living organelles like the mitochondria, like the ribosomes, like the endoplasmic reticulum, like the lysosomes. Similarly, there are also requirement of the some important chemical substances which comprises both the inorganic and organic, and all are this present within the cytoplasm, like lipids, etc., and several enzymes. Functions of cytoplasm. Here it helps in exchange of materials between cell organelles, the first function. The second important function, it is the site where certain metabolic activities also occur. Like here an example is given for you, that is the glycolysis. Actually the glycolysis is a process which is the first and the common phase of cellular respiration. During the process of respiration, when there is a breaking down, oxidative breaking down of the glucose molecule takes place, that will happen into the two different phases. The first phase is known as glycolysis, which occurs within the cytoplasm. Here is the partial without involvement of the oxygen, there is a partial breaking down of the glucose molecule takes place. And in the second phase, that after glycolysis, that is the Krebs cycle, where the process completed. That means complete oxidative breaking down in case of aerobic respiration takes place. Whether the process may occur in presence of oxygen, that is the aerobic, or the process if occur in absence of oxygen, that is the anaerobic. But this phase is compulsory and this phase is common for both the aerobic and anaerobic. And the third important functions, it helps in intracellular distribution of molecules, enzymes and nutrients within a cell. So these are some important functions. It is not only the revision. I have not summarized, but whatever may be the present within the syllabus, everything is here. Understand? Now we will move towards the second important part of within the cell, that is the cell organelle, that also includes the nucleus and the various other cytoplasmic organelles. First, we shall revise about the nucleus. Now, come to know about the nucleus. Let us see here a typical diagram of nucleus. In this diagram, the structure of nucleus, the nucleus surrounded by double membrane structure. May you can see here, this is the double membrane structure. That means nucleus is surrounded by double membrane structure. We will discuss the important parts of the nucleus, but before that, first we will know we will see the definition, the shape and the locations and the important parts of the nucleus in a very clear manner. Dear students, in this revision class, if you miss the things, then it is very difficult, as it is in a very detailed manner you have already studied. And now I would like to share with you the important points only. That is why you be careful in this regard. Like when we are talking about the nucleus is also a cell organelle. We are not talking about this structure, it is a cytoplasmic organelle, as because we have separated it. When we include the cytoplasm and the nucleus, then it is the protoplasm. And when we exclude the nucleus from the protoplasm, then it is the cytoplasm, and within the cytoplasm all the other different living cell organelles are present. That is why those organelles are known as the cytoplasmic organelles and also cell organelles. So this is the cell organelle, which is the largest cell organelle, and it is, what is its location? If you have an idea about the animal cell diagram and the plant cell diagram, then may you have seen there that in the animal cell, in the animal cell, it may present within the central part of the cell. Understand? But in case of the plant cell, it may present at the periphery, and that means not within the central part. That is why here it is written clearly, nucleus is the largest and the most important cell organelle in eukaryotic cells. You have also noticed here that I have mentioned here eukaryotic cells, as because you will know that a well-defined nucleus is not found in the prokaryotic cell. That is why it is here written in eukaryotic cells, located generally in the center of animal cells and at the periphery of the plant cells. What is its shape? It may be oval or spherical in shape. What are the important parts? There are 4 important parts of the nucleus. The first important part is here the double membranes which surrounded the nucleus. That is the protecting layer, just like that of the plasma membrane or the cell membrane for the cell, and for the nucleus there are presence of nuclear membrane. It is also known as nuclear envelope. Understand? And this envelope or the nuclear membrane is not continuous. May you have see here the pores. Here is the pore, here is the pore, here is the pore. That means it is not in continuous. And why this pore is present? It will allow the transportation of the substances from the cytoplasm to the nucleus and from the nucleus to the cytoplasm. As there are different types of things, substances are also very important for the other cellular part which are synthesized here, like we have been talking about the nucleolus. Then may we see that there are presence of the RNA and there are presence of the protein, and it helps in the ribosome synthesis. Now look, once again we come to our main point, that is what are the important parts. Very clearly there are 4 important parts. This is the first part, that is the nuclear membrane or the nuclear envelope, and the second important part is the nucleoplasm. Here nucleoplasm, that means this yellowish structure, that is this structure, may you see here, that is the nucleoplasm, just like that of the cytoplasm within the cell. Within the nucleus there are presence of nucleoplasm. It is also the matrix. And there are the third important part, that is the nucleolus. May you have see here, there are more denser than that of the nucleus. The nucleus is more denser than that of the cytoplasm, and here within the nucleus nucleolus is more denser than that of the nucleus, the other part, and that is the nucleolus. Here is the nucleolus. It is the third important part. And the fourth important part is the chromatin material, which comprising the chromosomes and the genes, that is the DNA and the other proteins. Here may you have now understood that there are 4 important parts of the nucleus. Depending upon these 4 important parts, this is a well-defined nucleus, and it is only present within the eukaryotic cells and eukaryotic organism. And as there is a no well defined, that means all these parts are not present within the prokaryotic nucleus. That is why they may be called the undefined and not well defined nucleus, due to the lack of the envelope or lack of the cell membrane. If there is a lack of cell membrane, then there is a mixing of the cytoplasm, and then there is no existence of the nucleoplasm. And that is, and only there are presence of this nucleoid, that is the chromatin material, that is the genetic part of the nucleus in the case of the prokaryotic. We may see it what is the structure of the nucleus in case of the prokaryotic cell, and may you have look here the eukaryotic cell. And nucleus controls all the metabolic. That is the important, the basic functions which I have written here in a single sentence. Nucleus controls all the metabolic and genetic activities of the cell, and that is why it is known as the brain of the cell. Now see here one by one the important parts and their important functions. One thing may you see here also in the diagram, this is the continuation of the nuclear membrane. There are presence of the endoplasmic reticulum. Understand? In continuation of the nuclear membrane, there is a presence of endoplasmic reticulum, and both the type of, maybe it when it in the diagram you may show in this way, the presence of rough endoplasmic reticulum, understand, and if you not put this dot marks, that is the indications for the presence of the ribosomes. That is why it is maybe look here clearly that if rough, then there is a presence of this dot like structure, and if it is smooth endoplasmic reticulum, it is without any this type of granules. Okay, it is clear now. Look here once again. The first part, nuclear envelope, are the nuclear membrane, which are the double and but not in a in continuity and not in continuous layer. It may be there are presence of several pore also. It acts as a barrier and separates the cytoplasm of a cell from the nuclear contents. It contains small pores which allow the transfer of selective materials from inside the nucleus to the outside. What will be here outside the nucleus? There are presence of the cytoplasm. That is why here it is written it allows the selectively materials transportation from inside the nucleus to the outside cytoplasm. You may have also seen here the matrix, the fluid parts of the nucleus, within which there are presence of the chromatin material. This is the chromatin material, intertwined entangled masses, which you thread like structure, which you may see here. That is the chromatin material, and this is the nucleolus, which without any membrane present on one side of the nucleus. Understand? Within the nucleus, without any membrane, there are presence of nucleolus. So here the nucleoplasm and the nucleolus. It is a liquid matrix within which nucleolus and chromatin material are embedded. Within which chromatin material and nucleolus are embedded. May you write in a simple way, within which the chromatin material and the nucleolus are present. It is the nucleoplasm. Now, nucleolus, a small spherical body which are present on one side of the nucleus is known as the nucleolus, and it is the part which are not surrounded by any membrane. Understand? And it is very important. It may be one or more in number and spherical in shape. Now, what is its function? The important function, it contains the protein, the ribonucleic acid, RNA, ribonucleic acid, and the protein, and it is helpful in the process of ribosome synthesis, and that is why it is also known as the ribosome synthesis factory. Understand? This nucleolus, I am talking about this nucleolus. All these parts which are present within this eukaryotic nucleus are not present within the prokaryotic nucleus. In the prokaryotic nucleus, you cannot see this one. That means the nuclear membrane. When there is no nuclear membrane, that means there is no existence of this nucleoplasm, there is no nucleolus at all, and only this chromatin thread, that is the genetic material, which is known as the nucleoid in case of the prokaryotic cell, is only present. Understand? And in this way we can differentiate between the prokaryotic and eukaryotic cell. Understand? Now, come to know about the most important part of this nucleus, that is the chromatin material, which may you see here in thread like entangled masses. Understand? Which at the time of the nuclear division became the chromosomes, and that is the chromosome, which is a rod thread-like structure where there are presence of the DNA, that is the gene, which is the hereditary material. Now look here, the last part, not the next, just we have talked about the three important parts: nuclear envelope, nucleoplasm, nucleolus, and the fourth important part is the chromatin material. The thin thread-like structure which you are looking here, intertwined mass of filaments present in the nucleoplasm. Just you have seen in the case of nucleoplasm, you have seen that the nucleoplasm is the matrix, the fluid matrix where the intertwined masses of chromatin materials and the nucleolus are embedded. That's why here it is written, the thin thread-like intertwined mass of filaments present in the nucleoplasm comprises chromatin materials. That means the nucleoplasm is the locations, the site for the presence of this chromatin material. Now I have written here 1, 2, 3 important points in regard to this chromatin thread, in regard to this chromatin materials, in regard to this nuclear reticulum, it is also known as. What are these 3 important points? Look here, the first one is here, the chromatin filaments. These chromatin filaments are composed of DNA and proteins. Okay, that means you may have seen here, you may have just see here in the previous part that in the nucleolus there are presence of the RNA, and here in this, within this chromatin thread you have seen that there are presence of DNA. That means the both DNA and RNA are here within the nucleus. DNA is the genetic material and contains all the information required for metabolism. All the information required for metabolism. Metabolism is not a single process. It is the process which comprising the 2 important phases, the constructive process and the destructive process within the cell. The constructive process means the formation of complex substances with the help of the simple substances, and again the conversion are the functions which may break the complex substances, and it may form into the simpler, from the complex to simpler, that is the destruction, that is the anabolism and catabolism. When there is a construction of the complex materials from the simple, that is the anabolism, that is the constructive phase. And when there is a destruction, that is the catabolism, that means conversion of complex substances into the simpler form, and this is the metabolism. And all the biochemical reactions, that means all the biochemical, bio means life, and the chemical reactions which occurring within the cell is the metabolism. Growth, reproduction and hereditary characters of an organism. This is the second important point in regard to the nucleus. The third important point here, during the process of cell division, the chromatin, this is the chromatin material which condenses during the process of cell division into distinct structures called chromosomes. And depending upon this, you may have studied, I have given you an idea in the general classes, that depending upon that there is a different structures of the different shapes of the chromosomes visible, especially in case of the metaphase stage of the cell division. You may look the shape. Now may just here I would like to give you an idea about the chromosomes. Just you have seen in the online classes the entire structure. Here is the chromosome. This chromosome is the rod-like structure, the thread-like structure, which are maybe constricted at some specific place, that the place where it constricted, that means it is just like that, this one, and there is a constriction here, constriction just like that, and here is constriction. This constriction is the centromere. This constriction is the centromere. This is the centromere. Now, just looking here, just that maybe this is the centromere, and here is the one part of the chromosome, and here is the second part of the chromosome. It is not the second part, it is actually the in continuation, but there is a constriction, and there are two arms which meet here at this place, at that the place is known as the centromere, and this arm is known as chromatid. Understand? In this way, you have also made an idea that depending upon the centromere, location of the centromere, where is the centromere located, the shape of the chromosome, the shape of the chromosome, the shapes of the chromosomes may be like acrocentric. You have studied this all in your classes. Just once again, I am writing here. The first one is the metacentric, and the second one is the submetacentric, and the third one is the acrocentric, and the fourth one is the telocentric. I have written here 4 different shapes of the chromosome. Now look the shape. Just you may talk about this one, metacentric. In this metacentric, may the both arms are equal. In the submetacentric, maybe one are just smaller, somewhat smaller than that of the, in case of the metacentric. In case of the metacentric, both arms are equal, and in case of the submetacentric, one arm is slightly smaller than that of the other. Here you may look very clearly. And in case of acrocentric, the one arm is very much smaller, whereas the other arm is very much larger. And in case of the telocentric, telo means end. That means at the end of the arm there are presence of the centromere. This is the telocentric. In this way, you just I have revised these things for you, as you in a detailed way. It is already discussed in the normal classes. Now after this, you may also, I also revise the cell division. Just in a very brief manner, may you look here, the cell division. Cell division means the division of a cell. And if you are talking about the cell, that means you are talking about the nucleus and you are talking about the cytoplasm. And that is why when we talking about the cell division, that means we are talking about the division of nucleus and about the division of the cytoplasm. When we talking about the division of the nucleus, that means the karyokinesis. Karyokinesis. It is the phase for the division of nucleus. This is the phase of division of nucleus. That means when a cell divide, from each pre-existing cell, the cell divide, and when there is no division of cell, there is no possibility of the growth, there is no multicellularity or organization. And not only that, for growth, development, but also there are some other reasons also for the division of the cell. Here the first phase which you have seen, that is the karyokinesis. Karyo means nucleus and kinesis means division. That means the division of nucleus. Similarly, after just division of the nucleus, there is a division of cytoplasm. So that is the cytokinesis. Cytokinesis. That means the division of the cytoplasm. The division of the cytoplasm. When there is a cell division, there are two important phases. The first phase is known as karyokinesis, during which there is a division of nucleus takes place, and the cytokinesis, during which there is a division of cytoplasm takes place. Understand? Division of cytoplasm. Now it's okay. Now look the different types of cell division. There are three important types of cell division. The first one is amitosis. The second one is mitosis, and the third one is the meiosis. When we're talking about the amitosis, that means we're talking about the division of the cell within the lower categories of the organisms, like that of, and in case of the some specific type of cells also, like in case of our cells, when our WBC cell, that means the white blood cells will divide, it will divide on the pattern of the amitosis. And when a nucleus and the cytoplasm of an amoeba divide, and after the division, it is not only the case of cell division in the amoeba, but it is also an illustration of the reproduction. Understand? That means that the amoeba is a unicellular organism. If their cells will divide, then there is no multicellular organization. Obviously, it will show the two different individuals after the division. In this way, maybe look here, the types of cell division. Types of cell division. The first one is the amitosis. The second one is the mitosis. And the third one is the meiosis. Mitosis is also known as direct cell division. It is also known as direct cell division. Actually, in a detailed way, not in your syllabus, it is not required to study in detail, but just I have may manage it for your understanding. A mitosis, that is the direct cell division. Why it is known as the direct cell division? As because in this process, when nucleus divide, they cannot undergo any stages. In case of the mitosis, when nucleus will divide, it will follow the 4 different stages, like the prophase, metaphase, anaphase and telophase. Such type of phases will not be followed during the division of nucleus in case of the amitosis. That is why it is known as direct cell division. Okay, the result will be similar in both for the amitosis and mitosis, as because during the process of amitosis also a cell divide into the 2 daughter cells, and in that daughter cells the number of chromosomes will be similar to that of the parent in both cases. And this mitosis is known as indirect cell division, indirect cell division, and also the somatic cell division, as because it takes place in the body cells or the somatic cells. That is why it is known as somatic. And why it is known as indirect? As because nucleus not directly divided, but it will follow the different phases or the different steps. Like that, what we have talked earlier, like that, prophase, metaphase, anaphase and telophase. That means there are 4 proper steps in case of the cell division. What are these steps? The prophase, number 2, metaphase. In this phase, the chromosome clearly visible. In this metaphase, at this metaphase stage, chromosomes will be clearly visible, which you have seen just earlier, that the different shapes depending upon the positions of the centromere, metacentric and metaphase, and the third one is the anaphase, and the fourth one is the telophase. Now come to this side, meiosis. Meiosis is also known as reductional division or germinal cell division. Understand? In this case, what will happen? There are formation after division of the parent cell. There are formation of the 4 daughter cells, and in all these 4 daughter cells, the number of chromosomes will be half in comparison to that of the mother cell in that of the original cell. That is why it is known as the reductional division, as it occurs in the reproductive cells. That is why it is also known as germinal cell division. Here, which you have seen here, the different 4 phases. In case of the meiosis, it is not only the limitations, but also meiosis comprising the two different. First, it will be meiosis 1, meiosis 2, and in meiosis 1 again there is a prophase 1, metaphase 1, anaphase, and prophase 1 is again divisible into the 5 different substages, and meiosis 2 only divisible into the 4 different stages: prophase 2, metaphase 2, anaphase 2, telophase 2. In the first phase, meiotic, there is a division, and also in case of the second, and altogether there are 4 daughter cells are the 4 nuclei will be formed, and in which the number of chromosomes will be reduced half in comparison to that of the parent cells. In this way, we have seen that once again may you carefully listen that the type cell division having the 2 different phases, the division of nucleus and the division of the cytoplasm. Whether there is any type, maybe the type amitosis, the type mitosis, the type meiosis, in all these 3 different types, that must be there is a division of nucleus and there is a division of cytoplasm, as because you will know that if the nucleus will divide, it will not exist without the supporting apparatus of the cytoplasm. Understand? And it is also known as some other terminology also useful for you, like the amitotic, mitotic and meiotic. Understand? It is also amitotic division, mitotic division, mitotic, and it is also known as meiotic. Understand? Also known as meiotic. Okay, so this is the nucleus and the important facts regarding the cell division. Now, in this revision session, we are in the last part. In the last part, we can summarize the important cell organelles. Here, the first one for you, like the endoplasmic reticulum. It is of 2 types, a smooth endoplasmic reticulum and the rough endoplasmic reticulum. When there are presence of the ribosomes, when there is attachment of the ribosomes with the endoplasmic ER, it is the rough. When there is no attachment of the ribosomes, it is the smooth. Characteristic features here in a very brief manner: network of double membranes found scattered in the cytoplasm, may be rough endoplasmic reticulum with attached ribosomes, or a smooth endoplasmic reticulum devoid of ribosomes. You have seen this diagram in the nucleus, and it also with the continuation of the nuclear membrane and with the scattered within the cytoplasm. Functions: provide mechanical support to the cell, serves as a channel for the transport of material from one part of the cell to the other. Rough endoplasmic reticulum facilitates protein synthesis. Smooth endoplasmic reticulum helps in lipid synthesis and various biochemical reactions. So, this is a brief summary of one of the important cell organelle or the cytoplasmic organelle, that is the endoplasmic reticulum. With the help of this endoplasmic reticulum, there are formation of the cell membrane or the plasma membrane, that is the biogenesis. That process is known as the biogenesis. Now, look about the second important cell organelle. Here are summary for the Golgi apparatus. It is also known as dictyosomes in the plant cells. In the form of granules, rods or filaments known as dictyosomes in plant cells. Secretions of cell enzymes and hormones, the important functions: modifies, concentrates and transport the synthesized substances. The very important here, the mitochondria. The third important are minute rod-like structure with double membrane, biochemical factories where food molecules are oxidized and energy is released. That is why it is also known as powerhouse of the cell. Understand? So, here we have seen in the summary that all the important metabolic activities, functions, whether it is the biochemical processes or any other synthesis or the secretions process, all are related with the different types of cell organelles. Now, look about some other important cell organelles. Now, look here about the lysosomes. It is a bag-like cell organelles bonded by a single membrane, contains digestive powerful digestive enzymes or tissue dissolving enzymes. Digest and destroy any foreign materials, digest worn out and poorly working cell organelles. And that's why it is also known as suicide packs of a cell. Now look here about the ribosome. There is no bonded membrane without any bonded by membrane. Smallest organelles contain RNA and many proteins, center for protein synthesis. It is the center for protein synthesis. Now look about some other cell organelles. Plastids. You will know that plastids are such type of cell organelles which are only present within the plant cells. One of the important cell organelles which are absent in the animal cell, that is the plastids. This is the largest cell organelles in the plant cells. Similarly, centrosome is such type of cell organelles which are not present in the plant cells. Plastids are of three different types depending upon the pigments. The first one is the chloroplast, important plastid, as because it possesses chlorophyll molecules which entrap the solar energy. That means it helpful in the process of photosynthesis. The second important plastid is the chromoplast, which provides color to the different parts of the flowers and the fruits. Maybe colorless or variously colored. Most common are green colored plastid known as chloroplast, bordered by double membrane and having their own genetic material, DNA, like that of the mitochondria. That means there are two different cell organelles having their own genetic materials, that is the DNA. The double membrane and having their own genetic material, DNA, like that of mitochondria. That's why I have written here like that of mitochondria. It means mitochondria also possess their own genetic material, DNA. Functions: imparts color to the different parts of the flowers and fruits. Chloroplast necessary for the process of photosynthesis, and leucoplast having no color, colorless plastid, store starch, oil and protein granules. Now at the last we have the two important points here. The first one is the vacuoles and the second animal cell and the plant cell differences. Look here about the vacuoles. Vacuoles are membrane bound fluid filled storage sacs. These fluid substances are known as the cell sap. These help plant cells to remain turgid and rigid due to the presence of cell sap. The presence of the fluid substances within the membrane bounded vacuoles are known as cell sap, and the membrane of the vacuoles membrane which are bounded by membrane, that vacuole membrane which surrounds the vacuole is also known as tonoplast. Here the difference between the animal cell and the plant cell, very easy point, and all the points are relevant to you. Like cell wall is absent in case of the animal cell, but the cell wall additionally present along with the cell membrane or the plasma membrane. The second important point, nucleus located at the center, whereas in case of the animal cell, nucleus is present in the periphery of the cell. If the vacuoles are present, it is smaller in case of the animal cell, and the vacuoles are larger in case of the plant cell. Centrosomes are present which help in the process of cell division. In the case of the plant cell, there is no centrosomes. Plastids are absent in case of animal cells. Plastids are present in case of plant cell. That's why animal cannot photosynthesize, whereas plant can able to photosynthesize. Smaller than plant cell, the size of the cell usually smaller than plant cell of the animal cell, but in case of the plant cell, it is larger than animal cells. In this way, we have revised the important points related with the chapter number 5, the fundamental unit of life. Thank you very much.